brain, let's get down into the spinal cord, which is going to be very similar to the brain, but there are some structural differences in particular that we'll want to take note of. So the spinal cord is enclosed by the vertebral column. It starts at the foramen magnum. When the medulla oblongata left the foramen magnum, it became the spinal cord. And it actually ends a lot higher than many people initially think. It's at L1 or L2, the first of the lumbar vertebrae. It's providing two-way communication to and from the brain and body. It's got a lot of reflex centers. There are some reflexes that need to go all the way up to the brain for processing. They can be initiated and completed at the spinal cord. Now, the information will still be passed up, but the reflex itself is held within the spinal cord. So it's protected by bone. Not quite as tightly as the brain was, but the meninges are still there, and cerebral spinal fluid is still there. The spinal dura mater is only one layer thick. Remember, it's not quite the same as it was in the brain. It doesn't attach to the vertebrae the way it attached to the skull. The epidural space is a cushion of fat and network of veins in the space between the vertebrae and the spinal dura mater. Cerebral spinal fluid is going to fill the subarachnoid space between the arachnoid and pia maters, and the dural and arachnoid membranes extend into the sacrum and beyond the end of the cord. The cord ends at L1 or L2. So you can stick a needle, should that be diagnostically or medically necessary, into that region and get a sample of cerebral spinal fluid in a process we know as a lumbar puncture or a spinal tap, a lumbar tap. So here is where that needle would go, into this space. Spinal cord terminates in a cone-shaped structure called the conus medullaris. There are filium terminae that extend all the way down to the coccyx. So those are fibrous extensions of the conus that are covered with pia mater, anchoring the spinal cord. There are also denticulate ligaments, which are extensions of pia mater that secure the cord to the dura mater. Cervical and lumbar enlargements are places that get a little wider. Those are places where nerves are going to the upper and lower limbs, the arms and the legs, coming off the spinal cord. The spinal nerves are part of the peripheral nervous system. They attach to the spinal cord with 31 paired roots. Each spinal cord segment is designated by the paired spinal nerve that comes from it. At the cervical and lumbrosacral enlargements, there are nerves, again, serving the upper and lower limbs arising there. And at the very end, there's a structure that looks like a horse's tail. So it is appropriately called the cauda equina, which is Latin for horse's tail. It's a collection of nerve roots at the inferior end of the vertebral column. Here's a picture of the vertebral column dissected away with nerves coming off. And a closer look at that. And down here we have the cauda equina. We have the segments of the spinal cord, cervical, thoracic, and lumbar below that. There are two lengthwise grooves that run the length of the cord, partially dividing it into right and left halves. That would be the ventral or anterior median fissure, or the dorsal or posterior median sulcus. The gray matter is located in the core, the white matter on the outside. So this is one of the differences between the structure of brain and spinal cord. On the cerebrum, we had gray matter on the outside, white matter on the inside, because the gray matter was talking to the rest of the body. Here, the spinal cord is going to be talking to the rest of the body, so the gray matter is going to be on the inside, white matter on the outside. Then the canal we've touched on several times, runs the length of the cord that is filled with cerebral spinal fluid. So we see our gray matter. Then we're going to talk about these horns and it's canal running through the middle. And another view. So the gray matter and the spinal roots, the cross section, as I said, looks like I called it horns. It does resemble a butterfly. You can also think of it as a letter H. And those horns that make up are the areas of gray matter found on each side of the center, and they are mirror images of each other. So the dorsal horns contain interneurons that receive somatic and visceral sensory input. The ventral horns have some interneurons in there and some somatic motor neurons that are going to talk to muscles. Lateral horns, which are only found in the thoracic and superior lumbar regions, are part of the sympathetic nervous system, so they contain sympathetic neurons. The gray commissure is a bridge that connects the masses of gray on either side and closes the central canal. That would be the body of the butterfly or the midline of the H. The ventral roots are bundles of motor neuron axons that exit the spinal cord. The dorsal roots are the sensory input to the cord. The dorsal root ganglia, more commonly called spinal ganglia, are cell bodies of sensory neurons. And the spinal nerves are formed by the fusion of the dorsal and ventral roots. Gray matter could be divided into 4 groups based on somatic or visceral innervation. So we could have somatic sensory, visceral motor, and somatic motor. The visceral motor would be part of the autonomic nervous system. So somatic sensory, visceral sensory. Visceral motor, somatic motor. White matter is myelinated and non-myelinated fibers that allow communication between parts of the spinal cord and the brain. They run in 3 possible directions: ascending, descending, or transverse. The ascending ones go up to higher sensors. They are taking sensory inputs to the brain. Descending are from the brain to the cord to the lower levels. Those are motor outputs. And from side to side, we have commissural fibers that are carrying information between neurons in the spinal cord. The white matter is divided into 3 columns, which are also called funiculi. On either side, there is a dorsal and a ventral, posterior and anterior respectively, with a lateral. And each tract is composed of axons with similar destinations and functions. And we'll talk about some of those neural pathways in the next section. So we have ascending tracts, descending tracts. Damage to trauma to the spinal cord can lead to localized injuries and lead to functional losses. Paresthesias are caused by damage to dorsal roots or sensory tracts. That leads to sensory function loss. Paralysis is damage to the ventral roots or ventral horns, and that leads to motor function loss. And there are 2 types of paralysis you should be aware of: flaccid paralysis and spastic paralysis. Flaccid paralysis occurs when there's severe damage to the ventral root or ventral horn cells. So in that case, impulses are not sent to the muscles. There's no voluntary or involuntary control of the muscles, and no muscle contraction, and that leads to the atrophy or the wasting away of the muscle. In spastic paralysis, there's damage to the upper motor neurons of the primary motor cortex, so that spinal neuron remains intact, and the muscles are stimulated, but not under conscious control. No voluntary control of the muscles. So they will tend to shorten, but they would not atrophy as much as they did in flaccid paralysis. Transection, or cutting the spinal cord at any level, is going to result in total motor and sensory loss of the region below that cut. If it's between T1 and L1, that would lead to paraplegia, and below or in the cervical region higher up, it would lead to quadriplegia. Spinal shock is a transient period of functional loss that would be below wherever that lesion is. Poliomyelitis is one of the, I'm sure you've heard recently, one of the epidemics that we've gone through since the turn of the century, well, since the 1900s. Poliomyelitis is a viral infection. The polio virus gets into ventral horn motor neurons and destroys them. That leads to muscle atrophy. It can also lead to death because of respiratory muscles not being stimulated, or cardiac muscle. And survivors often have post-polio syndrome, and that can happen years after the loss of neurons. ALS is also called Lou Gehrig's disease. It's the destruction of ventral horn motor neurons and fibers of the pyramidal tract. Symptoms include the loss of the ability to speak, swallow, and breathe. It's a very poor prognosis. Death usually occurs within 5 years of diagnosis. We don't know what causes it. There are a lot of ideas out there. It's probably a combination of environmental factors and genetic, and some of that may involve RNA processing. We know that glutamate plays a major role in the pathology of it. It's an excitatory neurotransmitter. And the only drug that offers any treatment at all for ALS is called Reluzzo, and it interferes with glutamate signaling.